Hi, welcome to the lecture on environmental adaptation for animals. Now we will consider on the circadian rhythm, so the daily rhythm. So how we can understand that, okay, is it day or night? And how we can maintain this rhythm even when we don't see the sunlight, or if the sunrise and is changing throughout the year. And then by modulating this circadian rhythm, then it's affecting that, okay, we can then have differences in the springtime, in the summertime, in the fall, and in the winter. So we can have this seasonal acclimatization. But let's start wondering first, okay, how the animal can decide that, okay, when it should wake up, when it's rested, but perhaps it also should wake up when if it's night active animal or day active animal, so it starts collecting food at a certain time of the day. So it must have some kind of mechanism to say that, okay, now it's the time to catch some prey. And we have pretty much everything is regulated with these daily rhythms. Of course, we have locomotion, so we are, for example, day-active animals, and so that's why we are walking more on daytime than nighttime. But also this wake cycle is affected, but it's also affecting on metabolic rate, body temperature, urea output and drinking, as well as in the circulation. So here you can see responses in human body that, okay, in the core body temperature is dropped about 0.5 degrees Celsius during the nighttime, and at the same time there is quite low urine volume produced, and then at the same time during the nighttime the cerebral in the brains, so that the cerebral blood flow is increased, and at the same time the cyclic pressure, so the blood pressure is low early in the night, and then high early in the morning. And the last one, it's actually having pretty drastic consequences, because it's changed about 2 times higher in the morning than in the late night. So that's why the heart attacks happens most often in the morning. Then it affects on the hormone control. So we have adrenocortical hormone secretion that we are going to discuss later during these lectures, but it's also affecting on several different hormones, of which in the daily rhythm we are going to discuss already on the melatonin. So the melatonin is not secreted in the daytime at all, and then at the night time there's maybe 200 times more melatonin. But it's also affecting a fluctuation in several other hormones: cortisol, tyrotrophin, growth hormone. They are all modulated, so either released at the nighttime or early in the morning. But then, beside these physiological parameters, we can have quite drastic consequences in different animal groups. So some fish and crabs are changing the color. They have different color in daytime and nighttime. Then the insect pupa, there's emerging adults at certain time of the day. And when you have an adult insect, if it's female, it's releasing pheromones. If it's a male, it has the ability to sense the pheromones, and then they can have mating, because it all happens at a certain time of the day. And also in very simple animals like paramecium, the mating is happening at a certain time of the day. And how to test this clock mechanism? We can do it quite simply. Test that, okay, what are the, for example, the body temperature or urea secretion in a long experiment. Over here we have in a human who was for 15 days without windows or clocks or computers, etc. And as you can see, the wake-sleep cycle is pretty constant. So he goes bed pretty much at the same time of the day. And this affects then that, okay, the temperature is changing, so there's this fluctuation. There's also fluctuation in how much sodium or calcium is released in the urea, or what is the volume of the urine. So it can be continued even in absence of daylight. And this is then possible only with some inner clock. And it can be regulated. So we have circadian rhythm. It means about a day, and it's kept in time with the sunlight. And for example, this scaffin, they are morning active. Very typical bird, for example, here in Finland, and they are singing quite a lot in the morning, and they are not only singing, but they are also hopping. And this hopping activity happens for a few hours early in the morning, and because they are day active, so the metabolic rate is also much higher in daytime versus in the nighttime, maybe 50% more. And okay, if we put these animals in constant time light, they can still have the rhythm. There's fluctuation in the oxygen consumption. There's this few hour burst of this hopping activity. But the interesting thing is that, okay, now the clock is fast, so they are not happening at the same time of the day versus compare these zeros in the bottom. So they are a little bit earlier and earlier when this animal is kept in constant dim light. So it's about a day, not exactly 24 hours. And it's in this daylight, for some reason, for the daylight, it gets the signal that, okay, to put the clock in the right time. And of course, it's good to have this kind of inner clock, because this animal is living, for example, in Finland, where before the midsummer there's constant sunlight, so it's very difficult to get understand from the sun that, okay, is it day or night. But what about then the night animals? Nocturnal flying squirrels, they start the activation exactly at the same time of the day, and this can be tested easily with an octogram. We measure the activity for a long period, and over here you can see that, okay, each line is a single day. So on the first line there's the first day, on the last line it's maybe the day 25, and every time these black marks, they present that, okay, the animal is active. They start pretty much at the same time, around 6 o'clock at the night. But if these animals are kept in constant darkness, then this clock is slow. So it starts the activity later and later and later and later, because it's in constant darkness. So this clock we have inside, it's somehow getting the signal from the sunlight that, okay, to put the clock in the right time. And that's why this sunlight is a phasing factor. It's called Zeitgeber. So it keeps the time of these inner clocks. So how the clock is working? It's pretty simple, but because in animals like humans, we have usually more clocks or some additional elements. So that's why it's making a little bit confusing. But let's have a simple one first. So we have some kind of activator that is causing the rhythm of the animal. And this activator is activating another molecule. And this activation is usually quite a slow process. For example, it increases the gene expression of this repressor. And then this repressor is inhibiting this activator. So there's a negative feedback. And okay, we could then think that, okay, now this repressor is inhibiting this activator, then this activator is not anymore activating the repressor, and then this repressor is not anymore inhibiting activator, and etc. So getting confused. But let's have a simple example. So the simplest circadian clock works with only 3 proteins. Over here you can see protein levels of Chi-C protein, and it has 2 forms, either with the phosphate or without the phosphate. And the total amount of this protein in a test tube is pretty constant for the 3 days, but the phosphorylation is having a cycle. So in the original picture in the top, you can see that, okay, the top line or the bottom line, they are thicker, and in the beginning the top line is thicker, then 12 hours later the bottom line is thicker, etc. So that's why the phosphorylation varies. So it's either it's phosphorylated or not phosphorylated. And then when comparing these phosphorylation, we can then measure that, okay, how many percent of this protein is phosphorylated at a certain time of the day. And you can see then that, okay, the maximum is maybe 80% and the minimum is about 30%. So there is a phosphorylation is about 24-hour cycle, and it's not in the animal, it's in the cyanobacteria. Pretty simple organism, and it's regulated with 2 proteins. So we have this Chi-C, which is binded by Chi-A, and now when having this Chi-A, this Chi-C is phosphorylated. This phosphorylation cause that there will be another protein that will remove the Chi-A, after which the Chi-C will be dephosphorylated. And then the merry goes around. And not only we have now 3 proteins that are interacting, but they also must affect on different things. And even in these Chi proteins, they are affecting on the packing of the DNA. So how compact the chromosomes are, and then it's affecting on the global transcription of different kind of promoters. There is a global gene expression and also expression of these 3 proteins. So there are more monomers of Chi-A, B, and C, and that keeps this merry-go-round. And this cyanobacter is using this clock mechanism to, when it's packing or unpacking the chromosomes, it's using it for the cell divisions. And interesting is that this cyanobacter is overcompensating it in cold. So you can easily put these 3 different proteins and ATP in a test tube and put the test tube in different temperatures, and then check that, okay, whether the Chi-C is phosphorylated or not. and it has about a day, 21 or 22-hour cycle. And in cold temperatures, it should work that every biochemical process is slower. And that's why we have this acclimatization. So in the chronic exposure to cold temperatures, we are having this partly compensation. So it's not as slow as it should be in for example, in acute experiments. But actually, this clock, this phosphorylation cycle in the Chi cycle is completely compensated or even overcompensated. So the clock is working a little bit faster in the cold temperatures. And the same thing happens also in the vertebrates. So we have individual cells that have circadian rhythms. Even in humans, we have in different organs, we have these rhythms. Usually we lose them if we don't get the Zeitgeber information from the main clock. But it can be found in all kind of cells. And even in fish, it can be found on every cell type. So in every cell type, there has some kind of light sensor mechanism for this daily rhythm. And this causes a light-sensitive bioluminescence in isolated fish cells. So when we are having 12-hour daylight, 12-hour night dark cycle, so the bioluminescence is completely in different level in when we have lights on and then lights off, and it's kept also during the light when having a constant darkness, but it's losing its rhythm. And actually, when it's been tested in a single cells, they have a rhythm, but they are not having the same rhythm. So that's why some of them are active at 1 o'clock in the night, and the other ones are active at perhaps at the 5 p.m. So that's why you can't see a nice fluctuation. But then, for these cells, if you give 15 minutes a light pulse, then during the 72 hours, 96 hours, you can see that, okay, all the cells are having this bioluminescence at the same time. So they are, again, synchronized. And how it works for example, in vertebrates. The basic idea is that we have proteins that are affecting on the gene expression. We have proteins like clock and BMAL that are working together to increase the gene activity of this period gene. And this affects that, okay, now from this gene we have messenger RNA, of which we will have pair proteins. And if these pair proteins are not binded by CRU or TIM proteins, they will break down. But with these CRU and TIM proteins, they are working together and inhibiting this clock and BMAL dimer. So then you have the cycle. So you have proteins that are affecting on gene expression, and the result of the gene expression is somehow inhibiting the activity of this gene. And pretty much similar it works in all kind of animals. In the fruit fly, at noon we have a protein complex with clock and SYK protein. They are inhibiting clock gene and activating PER and TIM gene. But because the TIM gene, it works a little bit slower, now this per protein is inhibited. Then later on, when we have more TIM proteins, they bind with the PER, and they are then removing these clock and SYK proteins from these activation sites of these genes. And then this clock gene is activated. So there will be clock proteins. And that affects that, okay, then in the down, these clock protein, there's clock proteins that are binding SYK proteins, and at the same time we will these protein complex with these 4 different participants will be inhibited with and one more protein crew that is affected by the sunlight. So a little bit complex, but still understandable. And still in the human, the problem actually is that we have some background or background processes, but it's still quite similar. So we have this clock and BMAL affecting on CRU and PER expression, and they are then affecting on different clock control genes. But the problem is that, okay, we have this negative limb is inhibiting its activation. And then the problem is that we have these kind of ancillary loops. So we have other loops that are also activated or inhibited, and they are activating or inhibiting this first cycle. And then we have another ancillary loop that is again activated or inhibited, and this is activating or inhibiting different clock control genes. And even these ancillary loops are interacting with each other. But the summa summarum, you have the clock and BMAL affecting on these CRU and PERs, and then you might have some sites or beside them. And these clock control genes are then affecting on circadian transcriptome, so you have different kind of protein expressions, etc. Thank you.